Hello, and welcome to ELISA Info, Boster's ELISA calculator. This video will give you a brief overview on how to use this tool to get the most out of your ELISA experiments. To start off, copy and paste your data into the box below. You can copy directly from your spreadsheet. Just make sure to not include any column or row labels, and make sure to place the standard samples with the highest concentration at the top of your data. In case you simply want to test out the calculator, you can click on the Demo Data button to automatically fill the box with dummy data. With the data grid ready, it's time to assign each of the wells to a specific sample. To do this, you can simply click and drag over the wells to label them. You can also click on them individually. You can alter the way the wells are placed by changing the different options found above the grid. The type of sample will switch the label type to either standard, blank, unknown, or control. You can also delete labeled wells by selecting the erase option. The start number dictates the number that will appear in the next label you will place. The sample fill direction determines if the sample number will increase vertically or horizontally. The replicates direction is similar to the sample fill direction, but for replicates. It will determine if the replicates will be placed vertically or horizontally. And finally, the replicates determine the number of copies that each sample will have in the specified direction. You can also click on the Clear Layout button to delete all labels and reset the grid state. Now it's time to set the concentration for each of your standard samples. For each sample that you have labeled as standard, you will get a new field where you need to input the concentration of that sample. Use the data from your experiment to fill in the fields. For a large number of standard samples, that could get tiresome. So you can use the Autofill In button to automatically compute the concentration of each sample. Just type the initial concentration, the number of folds, and whether to multiply or divide the initial concentration by the fold number. Don't forget to specify the units of your concentration in the standard concentration unit field as well. This will be later used in your report. Finally, you can fine-tune your results using the different method settings available. The experiment name will be used to identify your experiment in the report. The regression model determines the model used to fit your data. It can be either a 4-parameter logistic regression, or 4PL, a 5-parameter logistic regression, 5PL, or a simple linear regression. The most common model used is the 4PL model. The normalization method consists of a technique used to normalize your data. You can choose to not normalize your data, to set the maximum response as 100%, or to set the maximum response as 100% and the maximum response as 0%. Setting the maximum response as 100% will divide all of the response data by the maximum value, making your data have a maximum value of 1 and allowing negative values. Setting the maximum response as 100% and the minimum as 0% will map all of the response data to a range between 0 and 1. The plot settings allow you to change the axis scale of the plot. You can choose between linear, semi-log, and log-log plots. Afterwards, you can click on the Analyze button and wait for your results to appear. You can also click on the Clear All Settings button to reset all of the method settings. After a short wait, you should be able to get all of the results for your experiment. Specifically, you should see a chart with the fitted curve, its parameters, and a table that uses set parameters to calculate the concentrations of your unknown samples. It's important to keep in mind that the results that you got here are the result of an optimization process. As such, there is a chance that your results are only one of many possible and valid solutions. This is especially true for nonlinear models like the 4PL and 5PL models. Because of this, it may be very common to get completely different parameters even for the same model and data. With all of that in mind, you can now download your results as either a PDF or a CSV file. The PDF file will contain a copy of the results that you see on the web, while the CSV will contain the raw data found in the report table. That's it, so go ahead and try it out for yourself.